Good morning. Today I'm going to give a set of updates on things that we've been working on in the past several weeks, some readings relevant to some of the things we've been talking about in past meetings, and just basically doing a short overview of some things. So let's get started. So the first thing I'd like to highlight is Paki Bonchalia's work on her GSOC. She's one of our GSOC students. She's doing this work on applying neural developmental programs to the C. elegans embryonic data set. So we have this directory that she pushed called Growing GNN, and this is basically the code for a lot of what she's been doing in our meetings over the summer. And so the first thing we have is an interactive notebook, which has a lot of the code. So she's talked about this code, gone over it in the meetings, and so forth. We also have some files here, some Python files for the graph NCA. This is a next steps image. This is the data for the parent and the daughters. So the parent cells, the mother cells, and the daughter cells, and then the requirements for running this from the repository. So if you clone this repository, you can get access to this program, and you can run it. This is an example of a growing graph of the C. elegans embryo. So you can see that we start out with 2 cells and we start to increase our complexity as cell divisions occur. So up here is the embryonic time in minutes. And so these are the cells that result. So you end up with this layered network that is configured in different ways. These are basically the daughter cells and the mother cells present at any one time point in embryogenesis. So as you can see, that the network becomes layered, it gets more dense, and it's an interesting topology. Part of this is an artifact of the way it's rendered, so you have, I don't know if there's any significance to the ones at different layers here, or if they're just laid out in a way that maximizes their connectivity. But as you can see, it gets pretty unwieldy here at about 240 minutes. And this is about the time where we start to get the nervous system, the neurons that differentiate from what we call developmental cells. And so this is a really interesting set of results here, this graph. We also have the code for creating the graph. So this is graph PY. This kind of goes through initializing the graph. We have this class graph, which is a class representing a graph with nodes, edges, and labels. So that this code produces what you saw in the last image. Graph NCA actually does this by building a neural cellular automaton, and it allows this graph convolutional network to learn representations and update node features, and can grow or modify the graph dynamically based on certain rules. So this is implementing a neural cellular automaton for this. And so I think these two pieces of code run together. This is just an image of the 2-cell stage, P1 and AB. You can see that they're connected. And then, as we saw in the last image, we grow our network from this initial state of a 2-cell embryo. So we have this 2-cell state. We have 2 nodes, which happen to be sort of the anterior and posterior end, the AB being anterior, the P1 being posterior. And you have this simple connection between them, and then you start to get proliferation of cells in each pole, and you get the patterns of connectivity we saw in that image. We also have the CSV file, which features this relationship between a parent cell and its daughter cells. We'll often call this the mother cell, and then the 2 daughters, and then the birth time. So this is extracted from our existing data sets, where she's displaying this in a way that you can use to train the model, as we saw, and then get the results as we saw in those images. So you have P0, AB, P1, those are all parents or mother cells. So we saw from, for example, that example just in the last image where we had AB and P1 that were connected. AB and P1 are born from P0. That's the 1-cell stage. That's the fertilized egg. And then they divide into 2 cells, AB and P1. You have a simple connection there. That's at birth time 0 minutes. AB divides into ABA and ABP, so we have an anterior and a posterior oriented division. That's at a birth time of 17 minutes. P1 then at 18 minutes divides into EMS and P2. So you have this structure where you have these daughters being born at a certain birth time, and then the daughters can give rise to their own daughters as a mother cell. So P0 is the mother to P1, and then P1 can be the mother to EMS and P2. EMS can then be the mother to MS and E. And of course, as these cells become mother cells, they eventually are no longer in the representation. They can be removed from the representation. They now are represented by MS and E. And so one of the things we've had when we've been doing this stuff with embryo networks, we've had this problem of, what do we want to show? Do we want to show all of the cells in the topology, or do we just want to show the mothers and daughters? And so we could show all the cells and get like this hairball network, or we could just have the mothers and daughters and get these type of networks that maybe show a bit more structure and are a bit more relevant to the extant cells in the embryo. However, showing all the historical cells in the embryo actually give us some other insights that span division events. So for example, if we have A, B, and P1 in place, we know that those maybe can be treated as hypernodes, or they can be treated as sort of ancestral nodes that maybe condition the connectivity of those regions. And so you can see that this doesn't take into account cell migration per se. I mean, these cells migrate around the embryo, but just to show I think this is a distance-based connectivity metric where these connections are occurring, how you can organize them in terms of the topology of the network and the layout. So congratulations to Paki for presenting that to us as a pull request. It was accepted, and I look forward to her final GSOC submission soon. Okay, now I'd like to talk about a few papers on actomyosin dynamics. So we've been on this topic of actinmyosin and how we can have an actin filament network, and it behaves differently from just having actin filaments in isolation. And this is important because your cytoskeleton is formed from these actomyosin filaments, a network of filaments that form the soft active material. And so I dug up, I think we were having a discussion about it last week, so I dug up a number of references, and this is kind of what I found. So these are different types of actin-myosin models in the literature, kind of ways that people have approached modeling this problem. So the first one here is how actin network dynamics control the onset of actin-based motility. So I think one of the things that came up in the meeting was that we could use network theory or network science to do one of these types of simulations or models of the actin-myosin network. So usually the actin-myosin network is this physical network. People usually use some sort of multiphysics approach or some sort of biophysics modeling approach. But we can also use, kind of focus on the network dynamics or the network science aspects of it. So we can look at things like connectivity, we can look at things like centrality, we can look at things like network diameter and so forth. And putting those things in the context of function. So like if you have the network diameter shrinks as the physical properties change, that would be an interesting thing to understand. So this paper talks about how just general network dynamics of actin, how it controls the onset of actin-based motility. So I'll just go through the abstract of these papers and then leave it at that. So the abstract reads, cells use their dynamic actin network to control their mechanics and motility. So this is where the network is this interconnected actin filaments, and they're using this to control their mechanics. So basically the actin network will contract and expand, and it will move the cell. So the cell can move around. And of course, the cell will use this to move in certain places, to migrate, to change its shape, and so forth. These networks are made of branched actin filaments generated by the ARP2/3 complex. So this is a molecular complex that make up this branched actin filament network. Here we study under which conditions the microscopic organization of branched actin networks builds up a sufficient stress to trigger sustained motility. So this is where we have to have these stresses that build up that can trigger some sort of motility. So it's kind of like if you wind up a wind-up toy. How much do you have to wind it up to sustain its movement? So if you wind it up a little bit, of course, it walks a little bit. If you wind it up a lot, it walks a lot. You're putting energy into that system, and it's being released in a structured way. And what we're saying here is that in actin networks, you need to have some sort of stress that triggers sustained motility. So you have to put enough energy into it. And sometimes the energy, it isn't a linear thing, like maybe in the case of the wind-up dowel. It's a nonlinear thing. So you put stresses into the system, and then it allows it to maintain contraction, and it allows it to move accordingly. In our experimental setup, dynamic actin networks, or gels, so these are modeled as gels, these are modeled as rigid systems, are grown on a hard bead in a controlled minimal protein system containing actin monomers, profilin, the ARP2/3 complex, and capping protein. So this is a minimal protein system. I guess this is done in a gel, so they're isolating the actin network, and they're building this substrate for it, where they have these basic components of the actin network. We vary protein concentrations and follow experimentally and through simulations the shape and mechanical properties of the actin gel are growing around beads. So we have this actin network as a gel. It's being modeled within this the minimal components of the actin network in the gel, and it's behaved. This is a good analog for the actin network itself. Actin gel morphology is controlled by elementary steps, including primer contact, growth of the network, entanglement, mechanical interaction, and first production. So these are different. So first of all, you have these elementary steps where you have to have these components sort of in this order. So you have primer contact, growth of the network. So the network has to sort of, come into contact. These components have to come into contact. The network grows. Then there's this entanglement where they're, integrated, mechanical interactions. So then they start to move, and there are these mechanical interactions, and then finally force production. So if you think of a set of springs, for example, those springs would have to be sort of formed, and then they'd have to come into contact. They would, the network would grow as the springs came into contact and formed connections. Then there's this entanglement, which is the physical interactions of the network topology. Then you have these mechanical interactions, because as these physical connections are made, and then there's some mechanical action, there's this interaction that then can be both linear and nonlinear. And then finally, this results in force production, or the output of the network. And so in an actin network, if you stretch the actin molecules a certain way, this produces force as an output. But along the way, there are these mechanical interactions as a consequence of the topology. We show that varying the biochemical orchestration of these steps can lead to the loss of network cohesion and the lack of effective force production. We propose a predictive phase diagram of actin gel fate as a function of protein concentrations. This work unveils how, in growing actin networks, a tight biochemical and physical coupling smoothens initial primer-caused heterogeneities and governs force buildup and cell motility. So this is, we're looking at these properties of the network. There's this tight coupling. We have these heterogeneities that are smoothed over, and then we get this force buildup that can result in some sort of force production that is nonlinear. So this is an interesting kind of approach to modeling this using a minimal system. So minimal systems are often used to sort of get at sort of the essential components of a system. we'll have minimal cells, for example, because we don't want to model every aspect of the cell. We want to have the minimal components so we can understand how each component contributes to the whole. This is where we're using these minimal systems. So this kind of shows an example in figure 1 where you have these molecules sort of separated. So you have these filaments that are kind of disconnected and they're not doing anything here. Then you have these independent subnetworks that arise. So as these filaments start to overlay each other, interact, they form these subnetworks, which is of course interesting from a network science standpoint, because if we grow a network, we usually have disconnected components joining together. A lot of times when they join together, they form subnetworks, and then they eventually form a giant component or a fully connected network component. That is then, you can transmit forces from one side to the other, or you can measure the diameter and so forth. So you start with these isolated filaments, we get these independent subnetworks that are functionally independent but still form networks. Then you get these entangled subnetworks. So the independent subnetworks start to overlap. You then get this complexity here. You get these forces that are stored, and then you get the force output. So this is from A1 to B1 to C1 to D1 to E1 to F1. These are all images where they show, these are epifluorescence images where they show this process. And these are the animations down here to show how this complexity builds up over time. In B here, in 5B, we can see another example of this where this is kind of in the technical sense, not in the network science sense, but the first step here is primer contact or initiation of heterogeneity. So this is where we have the filaments that are separated and they're kind of on this membrane. Then you start to get biochemical heterogeneities where you get these overlapping filaments on them in different parts of the membrane. So this is where you have these subnetworks. Then you start to get what they have in this diagram are two phases of network entanglement. You have network entanglement and lack of network entanglement. In the former, we have smoothing of initial heterogeneity, so these subnetworks start to sort of get, they start to matter less as we fill in the gaps between the subnetworks and we form this large giant component. In the lack of network entanglement, or the latter example, we have amplification of initial heterogeneities. In other words, these subnetworks just continue to grow on their own or to continue to persist on their own. And they have a different functional, they have a different topology, but they have a different functional consequence. And so what we find from these sort of homogeneous networks, they're not really homogeneous, but they're a single connected component, is that you have these stress buildups. And so this enables some sort of force production through stress buildup. So you have stresses on the network, on the actin filaments. And as they sort of store stress as energy. And so eventually they will release this energy as a force. And so that's what they mean by stress buildup. It enables this sort of force production, especially nonlinear force production, that you might see in actin filaments and the results of these interactions and connections. In this other example, from this lack of network entanglement and this persistence of these subnetworks, you actually see a lack of stress buildup. So if you have these large subnetworks that are not connected, you have local force production, you have persistent heterogeneities, but most importantly, you don't have the stress buildup that results, and you get a different sort of dynamics from these networks. So I think that's a very interesting paper talking about how they're modeling this with this sort of minimal system, and they're relating this to the network dynamics. Of course, we have different ways of modeling these networks, so there are different sort of approaches and strategies that people use. So in this case, they're building an atomic model of the actin filament. So this paper is from 1990, so it's a long time ago. It's from Nature, and it's actually kind of an interesting paper because it kind of talks about this as actin filaments as an atomic model. So there are going to be different types of models we're going to talk about. The first is the atomic model. And so this just means that you have these, these atomic components that are sort of modeled, used to model the actin filament. So the abstract of this reads, the F-actin filament has been constructed from the atomic structure of the actin monomer to fit the observed X-ray fiber diagram from oriented gels of F-actin. So they've isolated F-actin in a gel. They've been able to construct its atomic structure, so they actually have it's a very reductionist approach to looking at actin filaments and their structure. A unique orientation of the monomer with respect to the actin helix has been found. So this is where they're looking at the X-ray crystallography of this structure of this molecule. The main interactions are along the two-start helix, the contribution from a loop extending across the filament axis provided by the molecule in the adjacent strand. There are also contacts along the left-handed genetic helix. So this is really a model where you can isolate the monomer, you can look at its structure, and you can get a sense of what's going on in that structure. So this is a very reductionist view of actin filaments. There's no network here, it's just what the nodes look like in this network. And so they give a lot of information about the molecular structure, its size, its chirality, and so forth. Then we can jump up to again back to the level of networks. In this paper, they talk about stress fibers being embedded in a contractile cortical network. So this again is going back to this network approach, but now we're talking about a contractile network. So we're not interested in the connectivity and the force production. We're interested in this network that's contractile. And so this kind of goes through this. Let's go through the abstract. Contractile actomyosin networks are responsible for the production of intracellular forces. There is increasing evidence that bundles of actin filaments form interconnected and interconvertible structures with the rest of the network. In this study, we explored the mechanical impact of these interconnections on the production and distribution of traction forces. So they're actually looking at the interconnections, they're looking at the mechanics of those interactions, not just the connectivity, and how traction forces, or forces that move the cell along the substrate, are produced and distributed. And these are produced throughout the cell. So this is not necessarily a homogeneous process. This is a heterogeneous process. By using a combination of hydrogel micropatterning, traction force microscopy, laser photoablation, we measure the relaxation of traction forces in response to local photoablations. So this is where you're ablating parts of the network, and you're looking at how the traction forces are relaxed and how they're not relaxed. So this again goes back to that paper where we had the subnetworks versus the full network connectivity, and we saw that the subnetworks didn't produce this buildup of stresses. The giant component connected network did, and so there was a difference there. In this case, what they're looking at is they're ablating different parts of the network locally to see what this effect has on traction forces. This is an interesting, again, it's sort of the same thing, but it's a little bit different because they're not using a minimal system, and they're doing some other things to it as well. Our experimental results in modeling of the mechanical responses of the network revealed that bundles were fully embedded along their entire length in a continuous and contractile network of cortical filaments. So this is where the mechanical response of the network revealed that bundles were fully embedded along their entire length in a continuous and contractile network. So we have to have this continuous network. It has to be contractile, but maybe not. Maybe it can be contractile more locally than in other places. But they're just basically showing this. We'll maybe go into some of the figures to see what this looks like. Moreover, the propagation of the contraction of these bundles throughout the entire cell was dependent on this embedding. So, we have this global contraction property. And, you can have contraction locally, but if it's not propagated throughout the network, then it's just kind of this local contraction. It doesn't really move the cell very much. It might move it a little bit. But to have this coordinated movement, you have to have propagation across the entire network, which means that there has to be this contractile property across the entire network, which in sort of implies some sort of homogeneity. In addition, these bundles appear to originate from the alignment and coalescence of thin and unattached cortical actin filaments from the surrounding mesh. So, we have these bundles. The bundles originate from sort of these thin and unattached cortical filaments. So this is actually kind of suggesting a little bit different topology than we saw in the first paper. So this kind of shows their elastic model. This kind of shows, so it's a little bit different than the other paper where they're using the minimal system. This shows their cortical meshwork. So this is a mesh network. This is a little bit different than what we saw in the last one. This actually has this structure where we have this meshwork. You have the elastic elements here modeled as a spring. You have these anchorage sites. You have these stress fiber meshwork connections. So you have this meshwork in the middle. That's this cortical structure. You have the stress fibers that connect to the upper and lower bounds here. These are so a stress fiber is an in-series elastic and contractile element. The elastic elements are here, and then you have this set of connections that are also elastic elements. So you see that you have this model is a little bit different. It's this mesh network that's connected to the edges, the stress fibers by these with a different topology. These stress fiber meshwork connections. You have anchorage sites on either side, which governs sort of how it moves around. So you have this sort of different topology that we're dealing with in this model. Now this is a Wikipedia article about a classic model called the Fences and Pickets model of plasma membrane structure. So this doesn't have so much to do with the cytoskeleton. It's just that this has the membrane structure of a cell, basically. So this is a classic model called the Fences and Pickets model. So we can go back and look and see how people have thought about the structure of this in the past. So the Fences and Pickets model of plasma membrane is a concept of cell membrane structure, suggesting that the fluid plasma membrane is compartmentalized by actin-based membrane skeleton fences. So this is what we're looking at with these actin filaments, and anchored transmembrane protein pickets. So again, we were looking at the actomyosin network. We're looking at the actin filaments. We have the skeleton, which are the fences. And those fences, of course, have to have pickets. So those are the anchored transmembrane proteins. So these are things that are going in and out of the membrane. And so these are, this is a little bit different than this network, but of course this network has to take this into account. As the network moves and gets displaced, it's not just, being able to do that in isolation. It has these, a lot of these different types of transmembrane proteins that have to be embedded in it. So this model differs from the older cell membrane structure concepts such as the Singer-Nicholson fluid mosaic model and the Safman-Delbruck two-dimensional continuum fluid model that view the membrane as more or less homogeneous. So these earlier models, of course, fluid mosaic model and the two-dimensional continuum fluid model, were sort of these homogeneous structures that were kind of the standard. And then people kind of came up with this idea that it's not homogeneous. It's not enough to be homogeneous. You need to have this heterogeneous aspect of it, where you have both the actin membrane skeleton and these transmembrane proteins in between. The Fences and Pickets model was proposed to explain observations of molecular traffic made due to recent advances in single molecular tracking techniques. So this was enabled by newer technology so that we can actually see what's going on in the network. So this is the membrane skeleton fence model. So the actin-based membrane skeleton meshwork. So this is based on a meshwork model. It is directly situated on the cytoplasmic surface of the plasma membrane. Membrane skeleton fence, or membrane skeleton corralling model, suggests that this meshwork is likely to partition the plasma membrane into many small compartments with regard to the lateral diffusion of membrane molecules. So this is where we're sort of, because of the function, it's becoming a heterogeneous network. And these membrane molecules, of course, diffuse along this network or along the structure. So the network isn't just this network topology. There are things embedded in the network topology, and there are also dynamics that are governing sort of the relative heterogeneity of this system. Cytoplasmic domains collide with the actin-based membrane skeleton, which induces temporary confinement or corralling of transmembrane proteins in the membrane skeleton mesh. Transmembrane proteins are capable to hop between adjacent compartments when the distance between the meshwork and the membrane becomes large enough, or when the meshwork temporarily and locally dissociates. So this is interesting because this kind of gives us this idea of, not only is it a heterogeneous network, in that there are things beyond sort of the immediate network topology we have to take into account, but that there's this compartmentalization. And of course, in modeling, we do this compartmentalization sometimes to deal with subsystems. And so this means that we sort of have to have this dynamic compartmentalization aspect of this network as well, and that it doesn't necessarily fit into the nodes and edges model that we were suggesting for the first two papers, or the first and the third paper. Finally, we have this last paper. This is a mechanochemical model of actin filaments. So of course, this kind of goes back to the second paper where there's just this straightforward sort of biophysics. In the first case, it's looking at the sequence and the structural biology of actin. In this case, this is a mechanochemical model of actin filaments. Not necessarily thinking about the network itself, just thinking about like its mechanochemical action. So this is from Biophysical Journal in 2012. So the abstract reads, in eukaryotic cells, actin filaments are involved in important processes such as motility, division, cell shape regulation, contractility, and mechanosensation. Actin filaments are polymerized chains of monomers, which themselves undergo a range of chemical events such as ATP hydrolysis, polymerization, and depolymerization. When forces are applied to F-actin, in addition to filament mechanical deformations, the applied force must also influence chemical events in the filament. So we have this relationship between sort of the structure of the actin molecules, the internal biochemistry, the forces that are applied to the F-actin, and then those forces transducing themselves into these chemical events, and then eventually the chemical events producing forces of their own. So, this is kind of thinking about this network in terms of, maybe the connections between them. Or we could think of the actin filaments as nodes. We could think of the actin filaments as edges, but those nodes and edges are going to have properties that are very different from, say, like our typical nodes and edges in a network, where they have chemical aspects, they have force production aspects, they have force absorption aspects, and so forth. We develop an intermediate scale model of actin filaments that combines actin chemistry with filament level deformation. So we have this actin chemistry on the one hand inside the actin fiber. We also have filament level deformation, so we have shape changes, shape deformations, and all those play a role in the network. So if you have these deformations, do those deformations propagate? Do they involve only one actin filament or many? Are they local or global? We also have changes in chemistry that hold the same properties. Are those unique to a single actin filament? Are they local changes? Are they global changes in the network? And how do they affect the network as a whole? The model was able to compute mechanical responses of F-actin during bending and stretching. So this is their model where they're actually able to get a mechanical response for F-actin during these different functional activities. The model also describes the interplay between ATP hydrolysis and filament deformations, including possible force-induced chemical state changes of actin monomers in the filament. The model can then also be used to model the action of several actin-associated proteins for large-scale simulation of F-actin networks. Altogether, our model shows that mechanics and chemistry must be considered together to understand cytoskeletal dynamics in living cells. So their approach here is not to model necessarily the connections between actin filaments or the sort of the biophysics of the structure of each actin filament, but to sort of take this intermediate approach that the mechanics and the chemistry are important, that the individual actin filaments, the structure within them and their sort of their functional attributes in isolation are important, but then that also, if you can simulate those, you can then build that into a network model and understand actin networks with more precision. One of the concerns they have in terms of modeling chemistry, modeling the sort of the force dynamics, is to look at sort of the changes in this network. So, they recognize that actin filaments are embedded in this network, but they also recognize that these filaments are constantly undergoing changes, such as polymerization and depolymerization, branching and severing fragmentation. So an actin molecule isn't a static thing within that network. It's always changing. And all the actin filaments in a network change in a similar way, although there may be heterogeneity there. So some might fragment, some might branch, some might polymerize at certain points in time. So you have to treat these sort of as their own thing, but they also have these properties that can be integrated into a network. The dynamic morphological change enables the network to remodel itself in response to external stimuli. So again, these components, these actin filaments, not only have sort of their internal dynamics, but they have this remodeling aspect. And so that's one important thing about the actin-myosin network, is that it's remodeling itself. It's not only growing and the dynamics are heterogeneous, but they also grow heterogeneously. They repair themselves. And so that's an important thing to incorporate into the model as well. Mechanical properties of actin networks also have been examined in a range of experiments, from single filaments, references 4 to 6, to networks of actin-associated proteins, or 7. So those are in the references. These experiments demonstrate that actin possesses unique mechanical and chemical properties, yet many of these observations have not been explained theoretically. In particular, a unified model does not exist, where the mechanics and chemistry of actin are considered together on an equal footing. So this is interesting because, we've talked about the mechanics of actin sort of generally. We've talked about the structure of actin, but the mechanical and chemical property coupling is something that's unique here. So, that's what we need. We need a unified model to have not only this network aspect, but a model where the chemistry is involved. And so this is where we get this mechanochemical model that demonstrates forces can have a strong influence on actin chemistry, and then that can result in this heterogeneity in the network. And so, of course, we come back to that second paper, the one on the biophysics, the structural biology of actin molecules, where they talk about some of these mechanical properties, and they talk about sort of the molecular structure of actin molecules and how this relates to its ATP state. So mechanical properties of actin filaments and networks have been studied extensively, in particular computational and experimental studies, and the persistence length of the actin filament have been performed. And so they talk about the average persistence length with all subunits in the ATP state being around 17 microns. The data show large variation ranging from several to a few tens of microns. So they kind of they parameterize a lot of the sort of the biochemistry in this paper. One of the factors that can affect bending stiffness is the nucleotide state of the subunits, ATP or ADP. From computational studies, FATP was found to be twice as stiff as FADP, and this is attributed to a structural change the DNase-1 binding loop in subdomain 2. So they're getting down to the structural biology of this. And so they're actually looking at the helical structure of F-actin and how that contributes to the mechanical coupling of bending and twisting. This is especially true for shorter filaments where it's more important. So these are different structural aspects that actually, you can identify and model and play a role in the force output. So actually they're thinking about this at two levels. They're looking at this at the atomistic level, and they're looking at this in sort of a more coarse-grained manner. So coarse-grained manner would be to just say, characterize a lot of these structural aspects as heterogeneities, assign some sort of parameter to those features, and move on. You can also have these more atomic models where you model every detail of the actin filament and you parameterize every detail and you try to model that. So there's this part in the paper where they talk about the chemical and mechanical properties of actin having been characterized in modeling studies in two different ways. A lot of experimental studies that provide rich information on chemical kinetics of actin and its binding partners. And this has paved the way for elaborate simulations shown in citation 42 and then 45 through 49. Mechanical behaviors of actin monomers and filament models have been studied with detailed molecular dynamic simulations, so ND simulations used for this purpose. However, ND simulations currently cannot access filament scale biologically relevant phenomena. So we have these detailed models and we have these atomic models that kind of give us a lot of really good simulation results, very precise, but they don't really give us the things that we need to know in terms of biologically relevant phenomena. In other words, we can model the actin filament very, in a very precise manner. But then when we start to fit it into a network and try to understand what it's doing, it's not as good of a model. So you can use molecular dynamic simulations to understand a lot about each individual actin filament. Then you have these coarse-grained models that can be proposed to model things like elastic rod theory that lose atomistic detail but gain this sort of biological insight. So the elastic rod theory has also been previously used to study F-actin buckling and force production, and filament severing by cofilin. These studies provided important insights into global F-actin behavior. And so this is something where, we have these atomic simulations that give us the great details of each actin filament, but we also need to use these coarse-grained studies to understand its biological significance. So I think that's enough on those. The reason I brought those up is because we've been talking about actin filaments, actinmyosin networks, the role of the cytoskeleton in differentiation, and then of course we have this other component where we have our models of growing neural networks, growing networks. We have the network science component. So if we can bring all these together, especially in terms of something like biotensegrity or some other aspect of cell differentiation, that's kind of where we want to go with this. So the final thing I'd like to talk about today is I've found a new resource on Glia and C. elegans. So a lot of times, because C. elegans is a model organism, they have a lot of resources that talk about different types of subsystems in C. elegans. And this one talks about Glia. So I'm interested in talking about this, especially in terms of how those relate to the connectome and C. elegans. So this book chapter is Glia and Invertebrate Models, Insights from Caenorhabditis elegans. So this really talks about Glia and invertebrates, but also, with respect to the C. elegans connectome. The abstract reads, glial cells modulate brain development, function, and health across all bilateral animals, which are the bilateral animals that we know, with a left and a right that are mirror images, more or less. And studies in the past 2 decades have made rapid strides to uncover the underlying molecular mechanisms of glial functions. So a lot of times when we model connectomes, we're modeling the neurons. We're sometimes modeling the connections, which can be axons with synapses, or we can have gap junction connectomes, which are the connections between the cells. But then we ignore a lot of the other things in the brain that are contributing to brain function or brain development. And so some of those things include glial cells, astrocytes, and some of the vasculature. And so we don't talk about those things, or even the intercellular matrix. So those are all things that we kind of ignore when we build a model with a connectome. And so one of the things here, of course, are glial cells. And glial cells, in fact, are maybe an integral part of the connectome. They support neurons. They have different, roles like modulating things, modulating brain function. And so that's why this is important. And so they go on to say the nervous system of the invertebrate genetic model, Caenorhabditis elegans, which we know, has small cell numbers with invariant lineages, mapped connectome, easy genetic manipulation, and a short lifespan. So these are the properties of why we use this as a model organism. Now, what's interesting here is we can then also understand the role of glial cells much more, in a much more enlightened manner, because we have a lot of the information that we need about all the other parts of the nervous system. These characteristics are revealing C. elegans to be a powerful experimental platform for studying glial biology. This chapter discusses studies in C. elegans that add to our understanding of how glia modulate adult neural functions and thereby animal behaviors, as well as emerging evidence of their roles as autonomous sensory cells. So, they're modulatory, of course. They interact with neurons and interact with neural connections. They also have some role in behavior, perhaps. And then they actually act as autonomous sensory cells, which I wasn't aware of before. But I guess they're sensing the environment of the nervous system. I'm not really sure what that means. Maybe they actually play a role in sensation. But in any case, the rapid molecular and cellular advancements in understanding C. elegans glia in recent years underscore the utility of this model in studies of glial biology. We conclude with a perspective on future research avenues for C. elegans glia that may readily contribute to molecular mechanistic insights into glial functions in the nervous system. So they have a list of abbreviations here, as we know, like molecular biology, other types of, cell biology has a lot of different types of abbreviations. So this is a nice overview of abbreviations. And so they kind of go into some of the aspects of C. elegans and how a lot of development and the function of development is conserved with other species, and this makes it another reason why it's a nice experimental model. And so there are a lot of things going on. And so they have a nice table here where they kind of show the glial properties and the glial impact on neuronal properties in the context of C. elegans, Drosophila, in this catch basin category, mammals, which of course is wildly diverse, but it's different from C. elegans and Drosophila. So we have this table key yes, maybe unknown, or no, and we have a lot of actually commonalities between C. elegans, Drosophila, and mammals, despite there being a lot of differences in a large taxonomic distance. So you have these glial properties interact with sensory cells, neurons, that's true of all these models, organisms. Interact with epi or endothelia, that's true of all the model organisms. Associate with synapses, NRAP axons, glia tiling. So the glia tiling, this is the organization of glia throughout the nervous system. Glia intracellular calcium dynamics, barrier function, very privileged /BBB spaces. Now in C. elegans, we don't know if that's true, but in Drosophila mammals, we know it's true. And all these other glial impacts and neuronal properties are all kind of shared between C. elegans, Drosophila, and mammals. basically maintenance, like engulfment of neuron fragments, stress response signaling to neurons, regulating neural aging, neural transmitter diffusion, synapse positioning, axon dendrite specification, neural pill positioning, axon guidance, and being able to act as a neural progenitor cell. Those are all shared amongst our model organisms. The two that aren't exactly the case is regulate neurons with single cell specificity. In C. elegans, that's true, and maybe that's because we have these sort of deterministic cell types that are generated. So we can have Glia that match up with a neuron, a specific type of neuron. We don't see this, maybe, well, it may be the case, but it's unknown in both in Drosophila and in mammals as a catch basin. So we don't know if neurons can be regulated with single cell specificity in these other models. We know it occurs in C. elegans, and so because C. elegans is such a tractable nervous system, that's been found to be the case. The other is trophic support for neurons. So in Drosophila and in mammals, trophic support means basically it supports its physiological state, the sort of the energetic needs or the metabolic needs of the neurons. And so that's what trophic support is. And so we can see that in Drosophila and mammals, that's both the case. But in C. elegans, it's definitively no. So there's no trophic support for neurons in C. elegans, at least from the standpoint of Glia. So this is interesting. This is the only one I think that's a definitive no in the entire table. So this is really interesting. So in the paper, they talk about the C. elegans nervous system and how it contrasts with other model organisms. So if we look at like the number of neurons in Glia in C. elegans, it's much smaller than something like Drosophila, where you have around 100,000 neurons. In humans, you have around 86 billion neurons and Glia. So this estimate is a little bit off, but you get the sense of the orders of magnitude difference. So very different sizes of nervous system. The human example, of course, is stand-in for mammals, so this estimate is a little lower, but it's orders of magnitude larger than C. elegans. Now, in C. elegans, of course, we have this invariant developmental cell lineage. So this means that you end up with cells of a certain fate. It's reproducible, and it always is the case. Now, in contrast to that, the development in cell-cell contacts of neurons and glia in other species are stochastic. So in C. elegans, of course, we have this mapping between neurons and glia. We talked about this with respect to this regulating neurons for single cell specificity. And so when you have a relatively small nervous system with this deterministic aspect, you can regulate neurons with cell-specific specificity quite easily. In most other organisms, we have this stochastic aspect. So in other words, there's this stochastic cell fate cell fate is stochastic, and this mapping between neurons and glia is also stochastic. So that's the reason for this, maybe unknown in this part of the table. Another aspect of difference between C. elegans and other model organisms is that neurons survive without associated glia, indicating that their survival does not depend on glial trophic or metabolic support. So this is why they say that trophic support for neurons is a no here in the table. And it's really because C. elegans neurons can survive without glia. And so this is, again, an aspect of having a relatively small connectome, having this sort of system of sort of deterministic cell fate. Importantly, however, loss of glial functions in C. elegans does not lead to structural or functional deficits in associated neurons. So this is why it's not neurons do not have to have glia. It doesn't result in any sort of deficit, and so this is all this is a difference in C. elegans nervous system. Is it because it's relatively small? Is it because of this deterministic cell fate? We don't know. But what the advantage of studying C. elegans does suggest is that we can understand glial functions at a very granular level. And so we can understand glial functions, regulatory functions, as independent of the confound of glial trophic and metabolic support functions. So another nice table is this table where they talk about C. elegans, Glia, associated neurons, and behaviors affected by respective Glia neuronal interactions. So they have these different organs. So you have the hook, the ray. These are both male-specific organs. Cephalic, outer labial, inner labial, posterior derid, anterior derid, phasmid, and amphid. They're also postcloacal, spicule. Those are all both male, and neuropil and mesodermal. And so these are all organs with associated glia. So some of the cells in our cell lineage tree, some of the terminally differentiated cells are actually in these different organs are actually glia. So AMSO and AMSO, AMSH are both glia. ILSH and ILSO and interlabial organ are glia, and so forth. We have, of course, those glial cells, and we have associated neurons. So those glia are associated with these other neurons here. And this is true of these for all these different organs. Number of sensilla. So usually it's 2, sometimes it's as high as 18. There's heterogeneity there. There are these documented functions of glial neuronal interactions. So, in this case, in the amphid organ, AMSH has a number of specific roles involving the maintenance of shape and plasticity, dendrite outgrowth, ion and pH regulation, engulfment of damaged parts, epithelial glial mechanobiology. And then animal behavior regulated by glia neuronal interactions include chemosensation, eustachian, mechanosensation, thermosensation, and pathogen recognition. So these are all important roles in the nervous system as well. So this is an anatomical map of C. elegans. This shows kind of the relationship between these glial cells and neurons and how they fit into these different organs. And then this is another nice diagram in figure 3 of these glial neuronal interactions in C. elegans. So this is the cross-section around the nose tip of some of these cells. These are these relationships in different parts in different organs, and another cross-section in F. So I think that paper on glia is a really nice reference. It shows male-specific structures. It shows hermaphrodite-specific structures, and it gives a nice view of the diversity of glial biology in C. elegans, and then it compares it with other organisms like Drosophila and mammals. So the last thing I'd like to talk about today is I'd like to congratulate Lukas Bastani on his talk during our Saturday morning NeuroSim meeting. This is the Saturday morning NeuroSim meeting from this last Saturday. And we usually talk about a lot of things related to neurobiology, neuroscience, biology. There are a lot of social science. There are a lot of things thrown into this meeting. And so Lukas has been working on the OpenWorm Studentship, and he's been working on his project, which has been looking at the proteomics of C. elegans and building pipeline tools to get proteomics data and bring it into a pipeline that we can use to model development. And then he presented on this paper, Comparative Genomics and Functional Study of Lipid Metabolic Genes in C. elegans. So he spent, I think, probably about 40 minutes on this paper, kind of going through the paper, each figure, each section, and kind of giving a really exhaustive overview of this topic. And so if you're interested in comparative genomics and lipid metabolism in C. elegans, this is a good thing to go over. This is on the Orthogonal Research and Education Lab YouTube channel, so please check it out. And check out Lukas's work. And so this is our DevoWorm repository. This is OW project. You'll find Lukas's work here, where he has these zip files with JSON formatted data sets for each of these proteins, and then a notebook that kind of goes over how to build pipelines from, how to extract these JSON files and use them. So that's all for today. Thank you for paying attention.